Yeah, thank you so much. That last talk is like, I'm so riveted by hearing other people's experiences because, when we started our immune therapy program, we were always hunting down the melanoma lung people and the liquid tumor people who have had the vast experience of that. And I got so much input that we now have like an endocrine service that's benign, but all they take care of are the IO toxicities, so that we can refer to them. We've had them come 3 times already to our grand rounds to let us know about how to manage these things. And if you haven't seen it, in JCO about 2 months ago, there was the 90-page document on how to handle those things, so I have a copy of that on my computer as well. So thank you again. I know I stand between you and the great evening, and I have a flight in 3 hours, but that's okay. So I'm going to get through this. This is my complex. So I'm going to, as was mentioned, I think I want to just present a little bit about what the Moonshot's about, what we're doing locally at our center in response to that challenge, and some of the aspects that come up with this whole concept of individualized therapy, because I think we all have a good concept of what that means. But as I mentioned to you this morning, the biomarker which is central to all this may not be the predictive biomarker that we all talk about, like you get a blood test or a tumor sample and you interrogate it and it tells you what therapy to go and you expect a response. It may not be that simple. So I love this slide. This motto, research cures cancer, I've had this for 11 years now. It was put out by the National Coalition of Cancer Research. They had made this their logo, and I really, like dorky kind of person I am, believe this. I believe that, we never move the needle unless we challenge the standard of care. It should almost be like a failure if we just stay where we are now. We've done good, no doubt about it. I'll show you how much we've done over the last 40 years. But we can't be satisfied with it because we're not curing patients, especially in the GYN tumors, right? So this is an integration between basic science, translational medicine, screening, and therapeutics, and they all work together. The more we integrate them and put people together outside of their silos, the more likelihood we're going to actually achieve this goal of making a lasting impact. And for those of you who put patients on trial, thank them for it. I do. I think it's something that even if they don't get a result, having them participate in the process is so central to the whole mission. So the Moonshot idea actually comes from JFK, who made this speech. You go ahead and click it. I'll run this short clip. This was at Rice University, which is just across the street from where we are. And he had this to say. Great. Do we have any audio?
Did he get shot? No.
Yeah, I know. Here? Yeah. Oh my gosh. Not everybody in Texas gets shot.
But why, some say, the moon? Why choose this as our goal? And they may well ask, why climb the highest mountain? Why, 35 years ago, fly the Atlantic? Why does Rice play Texas?
That's an inside
We
joke.
choose to go to the moon. We choose to go to the moon. We choose to go to the moon in this decade and do the other things, not because they are easy, but because they are hard. Because that goal will serve to organize and measure the best of our energies and skills. Because that challenge is one that we're willing to accept, one we are unwilling to postpone, and one we intend to win, and the others too.
Pretty inspirational. This obviously was our attempt to get to the moon before the end of 1970. So in 1969, we did reach the moon. As you know, many of the inventions that were needed to achieve that goal were made along the way. This is the concept for cancer too. We think it's that those technological challenges were probably easier to overcome. Certainly, we know this is the case because if you think about the Moonshot idea, this is about the 5th time we've tried to, have rebooted it. We've kept saying, well, it's like those people who predict the end of the world, and then the date passes. You're like, okay, well, it'll be in the next 5 years that we'll all be gone. Well, that's kind of what happened with the Moonshot. So this is an interesting story, and DeVita wrote a book about this, and I really encourage you to read it. It's something you can just sit down and run. Much because you'll recognize the names. Some of these people are still at MD Anderson, which is kind of interesting as well. But this is a drug called lymphocytin. It was actually a derivative of nitrogen mustard gas, and it was used on a cancer patient by a guy by the name of J.D., and he had a lymphoma, and you can see the lymph nodes in his head and neck and armpits. And he was given this, and 8 days later, this lymphocytin pretty much knocked out his cancer. And they kept it secret for 3 years because this was during World War II. And ultimately, Louis Goodman and Alfred Gilman, who you may recognize as the authors of the Goodman and Gilman Principles of Pharmacology book, both of them went on to be Nobel laureates, reported this after a 3-year embargo. And so this was then picked up, there was a lot of excitement when this got out into the community. And another military gentleman by the name of Dusty Rhodes, who at the time was the president of the Memorial Hospital for Cancer and Allied and Associated Diseases, said, hey, listen, we're on our way to curing cancer. So they put that curing in that sentence at that time. And our Congress was really interested in starting to support this. So it set up these 2 blue ribbon committees to advise the government as to whether or not this was worth the investment. And they said no and no. And so they did it anyway. And what they established out of that $5 million investment was the screening cell set that we still use today to look for new drugs. So fortunately, we had foresight, and this process continues on and continues to reap multiple amounts of benefits. But the real power of the quote-unquote moonshot came with the adoption or the funding of the National Cancer Act, which happened in 1971 by Richard Nixon. You can see him there signing. The important person in this is not our president, our to-be president, but this woman, Mary Lasker. She was her husband actually had died of cancer, and she was a very effective lobbyist. The story about what she did in Congress to actually get them to fund this thing was really amazing. I mean, she had set up these allies, and she was the socialite that made this happen. And so she was able to, through her influential lobbying, secure $100 million, which today is about $650 million for cancer research. This was the big step that went forward. Of course, they said, similar to Kennedy, that they would be able to do this in the decade, which was quickly breached. But it wasn't like we didn't learn our lesson, because Andy von Eschenbach, when he was at the NCI in 2003, said, okay, you know what, maybe not the end of the decade, but how about 2015? We'll cure cancer. So his director's challenge, which is in this document, which you can still read online, he basically says, I challenge you to cure cancer by 2015. And of course, that was blasted. And then we had this Prevention Act that happened in 2009, the ERA economic stimulus, which put money into cancer research. And then ultimately, in 2016, having passed that deadline in 2015, we had Joe Biden, whose son also died of cancer, then launched the U.S. Cancer Moonshot. So all in all, since we started this story, I put $100 billion into the mix. And you can see, this actually starts at 75 on here, so after the National Cancer Act was initiated, that all these rates have started to come down, as have the death rates. And this is kind of a telling curve, but these are the averted deaths based on the trajectory of new cases. So it's definitely had an impact, and even in ovarian cancer, disease which we often say is, this incurable situation, we've seen a decrease of 22% over the last decade and a half in incidence, and a decrease in actual death rates. But what's really missing here, and what most people don't realize, is that prevalence has actually dramatically increased. I just looked at this for this year. From last year to this year, there's been another 2,400 patients added to the estimated prevalence, which means if you were to sample in the United States, all the patients who have cancer, raise your hand, you would come up with 10 times the incidence, tenfold incidence. So how can you have decreasing number of cases, decreasing number of deaths, increasing prevalence? They're around a lot longer, right? So we're not curing them, we're not preventing them, but not dying as fast. And that's the value of all of these. And there's a great series of papers, which I didn't put in the talk here, but if you look at the approval for gemcitabine and carboplatin, when it was done against carboplatin, and you compare it to gem-carbo bevacizumab, those versus gem-carbo, you look at the gem-carbo arms of those two studies, they're identical, their PFS is the same. The overall survival, double in OCEANS as compared to the AGO trial. Okay, so definitely making a benefit, but when we talk about, well, PFS and OS, and those are our only endpoints in time, we're missing the story. This is the story. But, of course, we're not there yet, right? You can see here that liver, pancreas, and corpus cancer, actually the data that just came out just, I think, a couple days ago, shows that corpus cancer's death rates are continuing to increase. So we still have work to do. So as you start to think about how we can overcome these challenges and continue to make progress, we know that we have learned a lot. Most, what we heard about today, a lot about biological complexity. We know that cells are very screwed up, but they're not the only story, right? Their environment, the microenvironment, the lymphatic vascular systems, the stromal cells, and the immune system, they all contribute to this process. And so understanding those interactions between the the contribution of the individual components, genetics, pharmacogenetics as well, how well drugs work on an individual basis are all part of the story that we need to do. And so these provide a number of roadblocks. So this 21st Century Cures Act that Joe Biden pushed through was seed money. So while we think about this as being a big chunk of dollars into the system, it was really put in place to start the process. As I mentioned, the goal that has been put out there is that they want this to go by eightfold, that we want $8 billion to come from this one to actually affect some of the changes. And so these are the priorities of the Moonshot. So prevention or early detection, as you would expect, cancer immunotherapy in combos, genetic analysis of stromal and surrounding cells. So we've already got the TCGA. So moving beyond that is to look at the microenvironment. Enhancing data sharing, which I'll talk about, oncology centers of excellence, pediatrics, and this exceptional opportunities for Cancer Research Fund. Overall, the investment that the government's going to put in here is about 1.8 over 7 years, and as I mentioned, the goal is to get to $8 billion with private investment. So right now, the way this was set up, there was a blue ribbon panel that was put here. So maybe some of you, these faces you might recognize. There's James Allison, who's the person who's led the team that led to the discovery of CTLA-4, and Alfred Young, who's also at MD Anderson, who's done a lot of work with neuro-oncology, and you can see a lot of the very famous people that have contributed to this, to the mission. The funding was released last year. This is what I put this down here just so you can see what the website. It should be in the handout. This is if you're interested in collaborating. There's opportunities here. These are the RFAs that were first released, and you can see a lot of them are focused on cancer immunotherapy, which is rightfully so. So moving beyond TCGA, we really are looking at opportunities to take what we've learned about tumor and expand it into other domains. One of the things that's driving this is the fact that we have now better defined the endpoint, the end game. As I mentioned this morning, part of the ability to bring new stakeholders to the table is to have something that they could get for their investment. So part of that had to do with our interaction with FDA talking about endpoints. Many of you know that between 2006 and 2014, there were no drugs approved in ovarian cancer, 0. And so it was during this time period that we said, hey, listen, why is this the case since we can get it done in other tumor types? And their answer to us was, well, PFS is too difficult for us to understand in ovarian cancer, and you haven't shown us any situation where overall survival was improved. So we had this meeting, which lasted a full day. It was supported by advocates as well. And we had the advocates up there telling them what it meant to be alive with disease. And we spent a lot of time talking about how to make the assessments for response and PFS. And if you'll recognize, it wasn't too much longer after this that we were seeing the approval of olaparib for response. First drug approved for response in the end of 2014. So as I mentioned, we're having a follow-up meeting in June 14th, which is going to be focused on what you heard in the first talk, which is about the endpoints that are related to immunotherapy. So I'll be happy, hopefully we'll have another document that we'll get out of this, and we'll be able to improve the opportunities for us to look at IO therapy in ovarian cancer as an endpoint to engage new development in these sites. So we talk a lot about, this individualized therapy, and this is kind of the thought. You do the molecular profiling, you get the biomarkers that you need, you separate out which patients are going to respond to which, what color you are, you get the color drug that matches it, and somehow you're supposed to get an outcome that's going to be better. But one of the things that's really dependent in this process, kind of how like screening has these axioms, an individualized therapy that's using a biomarker actually has to have something that's identifiable that the tumor is addicted to. It informs its natural history, can be engaged appropriately, and will lead to a therapeutic benefit, right? These things have to happen. Now, these are easy to say and very difficult to achieve, especially in a disease like ovarian cancer, where you have no definable mutation to interact with, but multiple amplifications and deletions. Now, in the United States, obviously, you know this story, CML-AML, with the drug known as imatinib. This data came out in New England Journal of Medicine in 2002, and it showed that in these two tumor types where they expected less than 10% response rate, both their AML and ALL cases, that they had these response rates that were, or these lack of death at a very, very high rate. And when they looked at this in GIST tumors, they saw a very similar thing with a 5% historical of 5%, and you can see their overall long-term survivorships. So I call this like, making a goal on the first hit. And of course, it made it right into Time magazine, top medical journal in the United States in 2001. And you can look, new ammunition in the war against cancer. These are the bullets. Okay, so it was a great raging success. And so, as you know, right after this happened, just like we did back with the lymphocytin in the 1940s, we started using this in every solid tumor type because we expected the same thing. So, and it's clear that there are definitely these exceptional responders for which this type of relationship has been found. And so you can see Harold Varmus there, who launched an initiative to fund these exceptional responders. And we had a program at our center, still ongoing today, where patients that have unusual outcomes to targeted therapy are put into a registry so that we can understand more about them. And obviously, here's a case that led to an approval of a drug. This was a gentleman that was treated with melanoma that was treated on a clinical trial that actually led to the approval of this drug, which was vemurafenib. But that's not me. This time. And the study was stopped early. Right? So this led to a rapid approval. Here's another with a patient who had a large triple-negative breast that was treated with a PIK3CA. She had a PIK3CA and a KRAS mutation. It was treated with a MEK and an AKT inhibitor, had a dramatic response in 2 cycles. And a lot of these patients essentially, again, are being collected in a registry so that we can study them and figure out, why this type of unusual response hadn't happened. And I think overall, although we don't have the hard data that has done a randomized comparison between targeted therapy when there's a target present versus non-targeted therapy when there's a target present, hasn't really been done yet. The circumstantial evidence is that it does seem to pan out. And these are 2 studies I pulled that basically show that when you have matched therapy, and this is across a number of different tumor types, obviously probably doesn't play so much in GYN tumors, but if you look across the spectrum, in general, that seems to be true. And we're hopeful that trials like the MATCH trial will help to identify these and provide us with better understanding and better data as to whether or not we ought to do the matching or not. Impact 2 is a really cool study because it's actually the study that's set up to do this. I can tell you it's incredibly difficult to get patients on this study. But this is a study that essentially finds that you have a targetable alteration and then they get randomized to standard of care versus getting the drug. It's hard to do. It's kind of like doing a radiation versus no treatment, or chemo versus observation. Very hard to do. But the trial is actually set up to do it. The problem is it's a huge trial, right? You need a ton of patients, and so it's ongoing, but it's taking a long time to do. So as I mentioned, with when we started looking, we said, hey, listen, there are ovarian cancers that actually do have the targets for imatinib. And so we did a trial. This is a trial that was done at the NCI. We did the same trial at MD Anderson, and we got the same result, which is that it don't work. So we had no responses. We had a couple patients that actually stayed down for 6 months, but the median number of cycles was 2. That's because that's when we did the CAT scan. Okay, didn't work. So it's a little of this, and I think we're all guilty of it. Seeing the experience of immunotherapy and melanoma, and the first thing we did was to do it in ovarian cancer, expecting the same outcome. Right? So, and as we learned, there's this particular gentleman that I showed you before, on therapy, he developed a recurrence, and rapidly, and ultimately succumbed to it. So what we learned from this process when we start to biopsy these lesions to understand what the workarounds are, it's attacking it. So when I mentioned that on-treatment biomarker being important, the idea is that if we were to sample him here, and figure out what was going on in his circulation or in the tumor at this point, we maybe could have avoided this or put it off longer because we might have adapted our treatment to the tumor's response. All right, and now knowing what these bypasses are, we're now developing drugs to address that. And some of this is coming from our multi-omics platform. So this is also being supported by the Moonshot, where they're layering on in a single sample all of the genomic changes that can happen at the tumor level, the stromal level, the supporting cells, but then also the proteomics and the RNA, microRNA, RPPA studies, all these things being layered on top of each other to get a better picture of what the real story is. And these pathways that are showing the interconnections and the nodes along the way is a very dynamic process that is a response to treatment. So again, understanding what is happening in the tumor microenvironment in response to your treatment is going to be the way that we'll likely be doing our adaptive therapies as time go forward. So as I mentioned, the relationship between tumor burden, mutations, and copy number alterations you can sort out different diseases for how that relationship happens. Ovarian cancer, you can see over here, is mostly one of copy number alterations, and you can see it over here as well. And this is an example of what was shown in TCGA of the number of deletions and what's the other word I'm looking for? Amplification for all of these alterations through the genome. And here's a case, a patient of mine who you can see she's got all of these alterations that are amplified. All of these, no BRCA, but all of these amplifications. And P53 being mutated. So in endometrial cancer, we now know that there are several subsets that do fall in this space. I had a POL-E, I was telling Walter the other day, I had a POL-E patient who came in and she had 96 mutations, including 3 in each of BRCA1 and 2. It was really, really interesting. She had very undifferentiated tumor, relatively early stage. Completed her therapy, and she's very nervous about what's going to happen to her. But we know that these POL-E patients tend to have a very dramatic number of tumor antigen repertoire, and so we think that the immune surveillance would be quite high. And so far, the overall outcomes for that POL-E subgroup is nearly 100%. So even with very ugly-looking tumors, they can do quite well. So I'm not really sure that this is realistic to come up with this because we don't have a mutate. All of our tumors are not necessarily driven by a solitary mutation portfolio. So we have a combination of deletions and amplifications as well as mutations, and that really is going to inform what the real strategy will be coming down the road. And of course, if you look at ovarian cancer being a P53 dominant tumor, we know that the other cell types can be potential other opportunities. And for those of you who are following the low-grade world and clear cell, like Lillian, which we hope to get that trial open soon, is that we have these alterations which may be more frequent that we can actually use drugs that are already approved for other drugs, for other tumor types that can actually make a difference. And so in low grade right now, we're actually doing our very first trial, hopefully, of non-chemo versus chemo for frontline treatment. okay, based on the expression and response to estrogen receptor in that tumor type. So very exciting about that, and we now have a number of MEK inhibitor trials and combination trials in that space. The one story that I think is kind of driven is BRCA. We know we've talked a lot about this. We know these are active as single agents, but one of those categories I showed you was how do you take a homologous recombination compliant tumor and make it messed up so you can use these drugs? Okay, and so one strategy was the thought about using angiogenesis inhibition. This is Joyce Liu's data. It was published a couple years ago. As you know, there are 2 phase 3 trials, which one is already enrolled. Looking at this in platinum-sensitive and resistant ovarian cancer, the combination of cediranib and olaparib, compared to chemo. Okay, trying to figure out ways that we can make these this contextual synthetic lethality a reality for all comers. And one of the things that, I did show you is that one drug that's looking at CDK1/2. So that's in this space right here. But once we understand more about the complexity of the DNA repair pathway, these are providing us new targets to look at the wild-type patient and for whom these drugs may work. And so a lot of this work is ongoing. All of these drugs are in therapy trials right now. So lots to tell you about in the future. Understanding resistance is also quite important because as I mentioned, the ability to sample these tumors with circulating tumor cells is already available to us. We have a number of trials where we're capturing this. And one of the things that people will say is that drug resistance is equivalent to PARP resistance, certainly for platinum. But there's definitely mechanisms such as loss of PARP1 expression, this efflux pump where we don't see this relationship, and then newly described as this deficient non-homologous end-joining, for which we do have a discrepancy between PARP sensitivity and platinum sensitivity. So it's not going to be as easy as looking at their prior treatment and expecting outcomes. We're going to have to know more about these specific mechanisms and whether or not they're being leveraged by the tumor. And this is just an example of one of those pathways just recently reported last year that showed when the non-homologous end-joining pathway was deficient that these cells were able to survive. So I think there's some very interesting new pathways that are being discovered that would hopefully allow us to further tailor our treatment mechanisms. And this just came out in Cell Reports, very interesting. This is also by the Dana-Farber group, that's now showing that in tumors that have homologous recombination deficiency, so they are not restored, they did not revert, but they have microRNA regulation of the pathway, become resistant to PARP. So again, you dig deeper and you start seeing more stuff than you could ever imagine. So this is a process where the patient should respond because they got the mutation and we can show that they don't have the machinery to make it work and they still don't respond. Okay? This story, as I said, is getting complex. So what am I doing on time? At Anderson, we have a Moonshot also. Okay, 8 minutes. Okay. We have a Moonshot ongoing as well that's part of this. We've broken them up into these flagships programs. What I wanted to show you is this. Right now, our strategy for all patients that are surgically amenable is that they get scored with a laparoscope. So we'll take those patients to the OR. We'll take samples from 4 areas, the primary, 2 metastatic sites in the abdominal cavity and the omentum. And we take the omentum separately because we've shown in some previous work that the omentum actually is a tropic area for ovarian cancer spread. There are the neuregulin and the receptor, HER3, is present in that tumor tissue, which allows the tumor to have a better soil to grow. And so we are looking at that. So we take these 4 samples here. If they have a score less than 8, they go on to primary cytoreduction, but we are allowing these window trials. So these are up to 2 weeks of a single agent. Chemo-naive patients get operated on. Tissue gets resampled. So now we have pre and post exposure sampling in those areas, and then they go on to novel treatment or on to standard of care. For the patients that have very high scores, these are large tumor burden in the abdominal cavity, they get neoadjuvant, and they're allowed to participate on our either an induction therapy that's novel or a post-therapy that's novel. And we take, again, at the interval cytoreduction, they get tissue sampling if there's tumor available. So this allows us to assess our zero rates under novel therapy. It also allows us to do the paired match comparisons. And so we've been doing this now for about 4.5 years. We have another report coming out on how this is going, but we're quite interested in the immune surveillance for these. So we have 2 trials that are ongoing right now. This is Talozoprazole is a PARP inhibitor. We're looking at 2 studies that are running concomitantly. They're looking at induction of standard chemotherapy followed by surgery, followed by pembrolizumab combo with pembro maintenance. And another trial that's looking at another PD-L1 inhibitor, durvalumab, before surgery and after surgery. And so we're going to compare the samples here between those 2 studies to see what chemo does and what chemo plus IO does, and to see whether or not the microenvironment is altered. So this has been actually kind of a nice platform. Many pharmaceutical companies are interested in it now, and so the number of agents that we have available to us under these alliance and partnerships is actually expanding. So we're continuing to refine this process, and this is just an example of some of the things that we're thinking about as we go forward. So a lot of stuff happening. I've pretty much talked about this, but I do want to talk about the informatics piece of this in the last couple minutes. This is obviously tricky, right? Patient data on the web. I mean, if we all got if we have our so many pictures got hacked, you can imagine what could happen with getting patient data hacked. I don't know what EMR is available in Canada, but if it's one of the Epic or Cerner systems, all that data can be brought together. It does already right now. If you go and Care Anywhere on Epic, you can see what's happening in any hospital that has that particular platform. So it's already out there. The question is, how do we bring it together in a way that complies with the HIPAA guidelines, but then becomes also annotated and analyzable? That's going to be tricky. So the concept that's out part of the U.S. Moonshot is to leverage that data with individual patient data. For instance, if you have a specific, here's a patient who has a P53 mutation of a specific type, and if it's integrated across all of the patients that are potentially exposed to it, then that allows us to observe outcomes and treatment that have been used in a kind of a registry type of pattern. And big data allows the analytics of thousands, hundreds of thousands of patients to be done in very unique ways. So it's coming. And it's coming with stuff that I've got on my wrist right now, right? I got my Apple Watch on. It's already can tell me what my heart rate's doing right now and how late I'm going to be for my flight and all that. But they can take blood test, they can do blood glucose now. All that stuff's coming. And there are ingestible computers right now. Obviously, the cameras are available, but there are ingestible computers that will gather data in real time as it goes through your system. This stuff can be uploaded without your knowledge, actually, to some central repository, and all that data can be done in a very, in a seamless way. So regulatory environment around this is going to be important. But it's definitely coming, and so it's something to keep an eye out for. The new SAE that is not in the CTCAE version 5 is financial toxicity. We know that, at least in the United States, this is a huge burden, and it continues to increase. Tom Herzog and I did a survey of patients, about 1,400 of them, in a 3-week web survey. Couldn't believe the response we got. It was amazing. And we asked them, among many other questions, about how they felt about their impact of therapy. And we gave them a couple of scenarios. A treatment that improved PFS but without overall survival, or improved PFS OS but no cure. And we asked them, what would they accept? And you can see on this bankruptcy that 1 in 10 would accept, if there was no OS, would still accept filing for bankruptcy to get to treatment. So this is not sustainable, obviously. That's a lot of patients. 1 in 6 would take, even if they weren't cured, if they were going to live a little longer, they would take it all the way to bankruptcy. So it's something we need to be aware of, because there are definite negatives for that. These are 2 studies that were published last year I thought were very provocative. This first study was looking at pain scores, quality of life, symptom burdens, and they showed that patients that were filed for bankruptcy with the same efficacy had poorer outcomes for those parameters. And in the second study over here, patients that filed for bankruptcy had a hazard ratio of 1.79 for death. So Bankruptcy equaled death, increased risk for death. So pretty impressive stuff we're going to need to pay attention to. So a lot of concepts, right? Resistance, primary treatment, resistance, on biomarkers, endpoints, all this is part of the IOM. She's reminding me I got a flight to catch. This is part of the IOM. And this was a report that came out in 2016. It's available. It outlines the kind of the strategy. And this is important because this helps to inform how Congress is going to spend money. I love this. This is Omar Bradley, who was a general in World War II in Africa. He says, amateurs talk strategy, but professionals talk logistics. And really the logistics of precision medicine is all about these factors: mentorship, clinical trial lists, research networks, innovative trial design, meaningful endpoints, and the business of research reform. And fortunately, the new NCI director, whom we're going to meet with in about 3 weeks, made these his priority as well. So we're very hopeful that we can continue to carve a path forward, try to leverage all those things I mentioned, and to use this in an integrated way with the Cancer Moonshot. So again, thank you so much for inviting me. It's been a pleasure to be here. Thank you, Walter, and your program.
Do you have any questions for Dr. Coleman? Maybe I can start. So as a group, CCTG, the Gyne site, is trying to develop the next study in platinum-resistant ovarian cancer. What question would you want to be asking in that group of patients?
Gosh, that's so the question was, what question would we like to answer in the platinum-resistant ovarian cancer? I think that I showed you at least one strategy, which was the angiogenesis escape issue. I mean, I think everything that we've talked about, P53, IO, how does immuno-oncology therapy work in that setting? How do we overcome resistance? How do you overcome? All those are relevant questions. And I don't know which one's going to be the one. I think they're all going to be important. So, but right now, all of those are being studied in that setting. It's a very it's one where endpoints can be reached easily, but it's also one that has been littered with failed attempts. So I'm really about trying to find a good trial that's informative, and if it's informative, whether it's positive or negative, we still learn something.
Love to put you on the spot.
Uh-huh.
So you talked a lot about systemic therapy in viewing the future. How do you see the role of surgery in the future?
I love it.
What has defined us?
Wait till after ASCO. Did I say that? I think that it's a good question. I do think that there's still obviously roles for surgery. We have you can't cure pounds of tumor with just medical therapy. Certainly in our disease, that's not driven by mutation, right, or driven by a specific immune system feature that we can overcome. So I do think there's always going to be, at least for the foreseeable future, there's always going to be a need to establish diagnoses so that we can do the right thing, interrogate tumor, and then symptom burden relevancy. But I do think that identifying those patients and when to do it is really what surgery is going to ultimately we're going to have to figure out where to place it. This idea of just keep cutting and cutting and cutting is just probably not going to win. I think it's, and even if we do operate, understanding what happens in the response to surgery, we're going to have to understand that as well. So a great question that Diane brought up this morning about, why can't, or I think it was Diane, somebody who said, why don't we treat with anti-angiogenesis drugs right after surgery? You're right. I mean, that's when we have our big cytokine storm, and that would be great to be able to ameliorate. But its exact role continue to be defined. We got to study it though.